All right, so here we are, last lesson for the HL section of cell respiration. Yeah, now, a couple of things. So we are here at this point. We've collected a whole bunch of electron carriers. We've made a little tiny bit of ATP, but we are now going to see where the majority of the ATP in aerobic cell respiration comes from. It comes from the combination of these last two steps, the electron transport chain and chemiosmosis. Okay, so at this point, actually, I'm just going to go back here. At this point, glucose has done its job. We're not going to see this organic stuff anymore, because if you think about it, the yellow circles are representing the carbon, right? And once they join this cycle, they never really come out, right? They're now just like a part of this, and like some more gets added in, some goes away as CO2, but then it's now stuck here. And you'll see that once we move to ETC, the electron transport chain, we won't be talking about carbon anymore. We are only going to be talking about electrons and the electron carriers, and then finally ATP. Okay, so glucose has sort of done its job. It's either become CO2 and it's gotten rid of as waste, or it's joined this cycle and now it's just spinning around and constantly changing between forms of this organic, like 6 carbon, 5 carbon, 4 carbon, 6 carbon, and keeps going. Okay, so that's it. Now glucose is now, its role has been satisfied. So now we're interested in those 10 NADHs that we made and the 2 FADH2s that we made, which are going to go to the electron transport chain. Now, I want to show this, and while you are looking at this, I'm going to get ready to draw this on my own, okay? What you're looking at here is, or what I want you to focus on in this picture is, where are we in the mitochondria? Okay, so 1, 2, 3. All right, no, not like that. So what we're looking at here is, in here, mainly this part, this box here, okay? Because this is telling us the different parts of the mitochondria. The matrix, so that's the inside of the mitochondria, right? The fluid inside, sort of like a cytoplasm. You've got your inner membrane. This is the one that's super folded, right, into cristae. And then you have the intermembrane space, meaning out here somewhere is the outer membrane. So this next part, the electron transport chain, is taking place on the inner membrane. When we say on the inner membrane, we kind of mean across. So there's going to be things moving across the inner membrane between these two places, between the intermembrane space and the matrix. So this is not going out of the mitochondria, but rather between the membrane that's on the inner part of it. So everything's still inside the whole thing, but it's just moving across the inner membrane between the matrix and the intermembrane space. Okay? And what you see, what you're looking at here, is the electron transport chain. What is the electron transport chain? It's in this yellow box. It's a bunch of proteins, okay? And we know from the membrane topic in SL, the term integral protein, meaning that it's a protein that goes all the way through a membrane from one side to the next. And they are going to act as sort of transporters, okay? So they're going to accept the electrons from our electron carriers, and they're going to move them along. So really, think of it as like a sort of like a machine, or you know, those like one of those toys that kids play with like you put a marble in, and like there's a slide, and then it hits something, and then it like moves and does. So it's like that. It's like a sequence of like step by step by step actions that are going to take place. And each of these sort of proteins in the chain has a job. So it's going to pass from one to the next, to the next, to the next. So the main thing I want you to just see here is, where are we, right? Where are we in the mitochondria? Here you have our electron carriers that are in the matrix, right? They were formed by Krebs cycle and by, some were by glycolysis and some by link reaction, right? But they are in the matrix here. We're going to see that some stuff is going to get moved across back and forth. Okay. I want to, just before I keep drawing, or I switch to drawing, I want to jump to the end here. No, just kidding. Yes. Okay, this one. I want to show you this before I start drawing. Remember that all of this is dependent on oxygen being present, right? We've said that for aerobic respiration to happen, that whole meaning of this is that there's oxygen there. The first thing I'm going to draw on the other drawing, even though it comes like 8 slides later, is this oxygen. The reason is we want to give oxygen this name of the terminal electron acceptor. This, chemistry speaking, means that it's very, like, it has a high affinity for electrons. So oxygen is like very, very greedy. It attracts electrons very strongly. So in this case, oxygen is going to be at the very end of this entire chain. And it's the reason why electrons are actually moving along the chain. Okay, so I just wanted to give a bit of a preview of that. So let's switch to whiteboard. Okay, everyone can see that all right? Yes? Anybody out there?
Yeah, we can see.
Yeah, we can see
Okay,
it.
sweet, perfect. All right, so I'm gonna draw some of these proteins here. Okay, I'll just give them different colors. It's not really important what color they are, but all of these proteins make up what would be the electron transport chain. Let's say that this is the ETC, okay? And this green thing is a different protein. It's actually an enzyme called ATP synthase, okay? That's gonna be sort of a different part, okay? This will be for chemiosmosis later. Let's draw where we are. This is the matrix. This is the intermembrane space. And this is the inner membrane. And I wanted to draw my oxygen being over here. Okay, it's just hanging out at the end of the electron transport chain. Are we ready to do this thing? Here we go. Okay, so
What did you say the green thing was?
it's called ATP synthase. I'll write it and I'll get rid of it so you can see. Oops. Synthase. It's an enzyme. Synth means to synthesize or to make. So it's what after we do the first step, it's what's going to make ATP.
Okay.
It's an enzyme that makes ATP. All right. Let us begin the journey. All right. Our electron carriers that were made at various stages from all those reductions that we did to them. Okay, they are going to come here and they're going to donate their electrons. Okay, so NADH is going to come here to this first protein in the chain, and it's going to give up some electrons. Okay, so it is going to be, if it's losing its electrons, what is happening to NADH?
Aiming.
Mr. Nan?
Yeah,
Mr. Nan?
yeah.
Just a quick question. As it's confusing me for a long time, how about that extra hydrogen ion?
Coming. I'm about to mention it.
Okay.
Okay. What is the term? If NADH is losing electrons, it is
Oxidized.
it is being oxidized, right? So NADH gets oxidized here. In fact, that's why this whole process, like you will see in the PPT, this whole process is called oxidative phosphorylation.
Wait, oxidize is when something loses and another one gains, and then reduction is gain and then lose.
Yes, but the thing that loses is the one that is oxidized, right? So in this case, since NADH is losing something, losing the electron, NADH is oxidized.
Oh, so it's not a matter of like which comes first. It's just what happened. Okay.
It's to whom does it happen?
Okay.
Okay. So in this case, we're going to see that all of our electron carriers are going to get oxidized, and in the end, we're going to phosphorylate some ADPs to make ATP. All right, so NADH gets oxidized, and it's going to give its electrons to this first protein here. And I'm going to draw my electrons with, how about orange? No, let's go with yellow. It's more easy to see. Okay, it's going to give away its electrons to this this first protein in the chain. Okay. Now we said that over here you have oxygen that really likes electrons. It likes to pull electrons. So the sum of what's going to happen is these electrons, and I'll get rid of this in a second. These electrons are going to fall towards oxygen. Okay, they're going to fall towards oxygen, and as they do that, every time they get passed from one, and there are some other proteins like kind of hanging around in here, like there'll be like a protein that helps to move this over here and a protein that helps to move this over here. We don't need to know all those details, but as these electrons move from one place to the next, and this place to here, and eventually to here, every time they move along the electron transport chain, they lose a little bit of energy. Okay, they give away a little bit of energy. So they're kind of falling downhill, if you will. So they're at like a higher energy state when they're given away, and then oxygen pulls them towards itself. And as it's doing that, energy is released. And we are going to use that, the electron transport chain is going to use that energy to do something very important. As these electrons start falling, it's going to be able to, and here we go, Richard, this is where this becomes important. As a part of this reaction, there's like H pluses that got made, right? Like loose protons. The energy that's lost as the electrons move along the chain is going to help us to pump.
Why is there a frame rate counter? Oh my God, my Zoom is crashing 24/7.
24/7.
This is the 14th time I opened this stupid in just 10 minutes.
I'm sorry to hear that. Anyway, so as the energy is given up, as the electrons fall towards oxygen, that energy is going to be used to pump hydrogen ions or protons across the inner membrane into the intermembrane space. Okay, so again, and here's where, again, like the question that Richard asked, we made, every time we reduced NADH, remember it was like NADH plus H plus. So there's a whole bunch of these just kind of like hanging around in this area, right? So at this point, those protons and some of the ones that are given up, like the one that's actually attached here, those are going to come into play. So again, as this electron then passes from here to here, we're giving up energy, right? The same ones from over here. So then more protons, boom, can be pumped across here. You also have, FADH, one of the other, FADH2, one of the other electron carriers. It's going to give its electrons at the second protein here. So it becomes oxidized back to FAD. So you've got a whole bunch of protons that, again, as it does that, the H plus and the electron, right, separate. So you have another electron here, or two electrons that are going to fall towards oxygen. That released energy will help to pump these protons up to here. And essentially, what we're doing is we are constantly pumping protons, and I know I've used two colors, I wish I hadn't, but they're the same thing, purple and green are the same thing. We're using the energy from the electrons moving along the chain to power lots of H pluses moving into the intermembrane space. So if there was a goal for all of this, the goal is to get to get a high proton concentration, proton meaning H plus ion, hydrogen ion concentration, in the intermembrane space. Now, a DT, no, not a DT, Yushwan, where does this and this go? You had this question the other day.
They go back to the, I don't know, the previous processes.
Which we can call the something cycle. Krebs cycle, or to the link reaction, or to glycolysis, or to whatever. So this is where
the try something. Never mind.
But they're basically, they're going to go and get recycled. So they're going to go and get electrons again and then come back. Okay. So what we are doing here is goal number one. Create high H plus. Oh, I'm too close to the edge here. H plus concentration, right? So there's going to be a huge buildup of H plus here. Yeah? Before we go any further, does anyone want to ask any questions?
Yeah, we create a high H plus concentration in the intermembrane. That's
Correct.
what we want to do.
That's what we want. So I'm going to do a little, let me just go here. So where we've created this, I can go back, don't worry. We have created this H plus concentration in here. Right? So the H pluses actually went across the inner membrane like this.
Oh, okay.
Right? Because this is the matrix. Right? This is the matrix in here. Okay. So where have we created the high concentration? It's in the space between the two membranes. Okay. Okay. which we will mention this at the end, but if you think about it, it's kind of easy to do because I didn't do a very good job of drawing this just now. But really, the space here is very small, right? Like it wouldn't take very much to create a high concentration, right? So because the volume is small, you can easily create this very high concentration by pumping a bunch of H plus into a small space. So it's one of the structure and function links is the intermembrane space is very small, therefore you can rapidly create the gradient and also maintain the gradient very easily because it's a small area. Okay. That is the first stage. So that is the electron transport chain and the role of the electron transport chain to accept the electrons from our electron carriers, to pass them one to the next until they reach oxygen, and by passing them along and allowing energy to be released, using that energy to pump H plus ions into this space. Okay. Can anyone predict? What do you think? We have seen this kind of scenario before. What do you think is going to happen next?
Diffusion.
In a sense, yes. In a sense, yes. Where? Give me some directions. Give me what you're referring to, Richard.
Different from the intermembrane space to the matrix through the ATP thing.
Okay, so again, if you watch the animation, you would know this, is that, yeah, it's like we've created this gradient. We've created a high H plus on one side, low on the other. So H plus is going to want to go back, like it's going to want to go to where there's less of itself. Unfortunately, H plus is not very good. It's not, let's say it's not allowed to just pass through the membrane, okay, because it's charged and that causes a problem. The only way it can go back to the other side is through ATP synthase. Through ATP synthase. So yeah, we have a gradient, we have a high to low gradient, but we're only going to allow these ions to go through this one enzyme, okay? Now, as this ATP flows through the enzyme, think of it sort of like hydroelectric power. As water, if you have a waterfall, I think you did this in like grade 7 and 8, right? It spins like a water wheel or whatever, right? And you can generate electricity because that thing is spinning, which can then turn a turbine and blah, 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 right? And you can harness energy like this. So this flow of H plus ions through ATP synthase, and this is why I wanted you to see the animation, is the ATP synthase is actually going to it actually turns sort of like a spiral. That's really poorly drawn. It actually turns like this, okay, in 3 dimensions. So as H plus ions flow through, that should be in green, sorry. As they flow through, it helps to turn this ATP protein, ATP synthase protein. Can I take a small shift and switch to this? Okay, so just to show you what it kind of looks like, you don't need to hear all this stuff because it's a little bit too intense, but I just wanted to show you what actually happens to the enzyme as hydrogen flows through, if it will ever play. What? Okay. I think maybe. So this goes into way too much detail about like the structure of ATP synthase. We're not interested in that. I just want you to see that it turns, like physically this whole thing turns. And come on, oh, here we go, sort of. Thinking about it. Okay, maybe
So
I need to
the
let it
oxygen
buffer a bit.
accepts the electron.
Okay, I'll come back to this. Never mind.
Mr. Nilan.
Okay, well, we're back to ATP synthase here. Okay, so what this is going to do, sorry, what this is going to do, I thought I erased this already. As the H plus flows through ATP synthase, is it's going to power ADP and like phosphates that are just hanging around to get shoved together to make ATP. So this spinning ATP synthase is going to take ADP and hanging around phosphate, and it's going to shove it together in there. And I wish the animation had played because then you could see it actually happening into dun, da, 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 ATP.
What just happened? I just came in and I see a bunch of H pluses.
And if we keep ions, protons flowing through ATP synthase, we can keep ATP synthase turning, which means we can keep mashing ADP with P to make ATP.
But where does the ADP and
As
P
a
come from?
final step, O2 is over here hanging out, right? In order to maintain a high H plus gradient here, we need to make sure that this side, the matrix, is relatively low. But now we have H plus coming down to this side, right? It's coming through into here. Well, that's going to balance everything out. If you balance everything out, now there's no reason for H plus to flow, and that's going to stop the production of ATP. Well, what can we do? O2 is getting 2 electrons from here. 2 electrons plus, if you add 2 hydrogen atoms, guess what you end up with? H2O, which is the end product of cell respiration, CO2 plus H2O. So O2 grabs electrons from the electron transport chain. It grabs some of the leftover hydrogens on the inside, takes it for itself, forms water, and now we're cleaning up the matrix so that the intermembrane space stays high, H plus concentration. We remove these out of the equation, anything that doesn't get pumped across, by coupling it with O2 to make H2O.
Wait, Mr. Nilan?
That process is called chemiosmosis. Okay, the movement, osmosis. Richard used diffusion. For some reason they didn't like chemi-diffusion. They termed it chemiosmosis because I guess it's going across a membrane. I'm not really sure how the naming came about, but the movement of those protons through the membrane through a protein to power the formation of ATP is called chemiosmosis.
Mr. Nilan,
do you explain?
And that is where we eventually get up to 32 additional ATPs from all that. Remember, we have 10 NADHs, right? 10 of these, and we have 2 of these. So for all those electrons that get brought in, we're constantly able to power ATP synthase to keep turning. I'm gonna let that sink in for a second.
I have no idea what you just said.
Mr. Nealon.
Wait,
I'm going to switch to the PPT in a minute and just go through it all one more time.
what?
Jay, check your phone.
Are people talking?
Okay, great.
No, right?
Yes.
Yeah, we are talking. You
Wait.
can hear us.
I can.
Yeah, but he can't hear me.
Really? Okay.
I don't know why.
Wait, Mr. Milan.
You're good.
I think he can't hear any of us.
Oh shit.
What?
Mr. Wait. Oh, let's a second. Because like when William came in, right? He was like, I don't know what fucking happened, and he didn't say anything.
I think he can hear us. Like Mr. Nealon.
Mr. Nealon.
Mr. Nealon.
Got you. I got you.
Oh, can you hear us?
Yes. For some reason.
Okay.
For some reason. Oh, I think when I was starting to play this animation, I turned the volume off. Okay. Sorry. I'm here. What's up?
Is the ADP and like the PI always in the matrix, or like where did they come from?
Yeah, they're always just around because they would have been used for something else and broke, like ATP broke apart, and then yeah, it's just, we can assume that they're just floating around. Same as how it got made for in Krebs cycle.
Mr. Nealon, can you really quickly just sum that up again?
Yeah, I'm gonna go through everything one more time.
Okay.
But like for the formation of H2O,
Mhm.
like how many hydrogens and electrons are like connected to one molecule of O2?
Okay, so I'll do all of ETC, and then we'll pause, and then we'll do the chemiosmosis, and then we'll pause, and I'll talk about the water again. Okay. So here we go. So we're starting here. We've got NADH, which shows up, gives away, we split up a proton and the electron, right? And this electron is going to travel and get passed along to one to the next to the next. Okay, it's going to travel across from pink to yellow to red, for example. That the energy that's released as it drops, or as it's being passed along, is going to pump the proton that broke apart, right? Because when you give away this H plus, an electron also comes off. So those two were together, now they're apart. So that gets pumped into the intermembrane space. So that gets passed along, eventually ending up over here where you would have O2 that's sort of drawing the electron towards it. And as you do that, each time a little bit of energy gets released, we use that energy to pump another and another H plus ion into the intermembrane space, slowly building up a high concentration of H plus. Okay, NADH gives its electron to the first protein in the chain. FADH2 gives its electrons to the second protein in the chain. Not really super important. Also, after they are oxidized, they return back to Krebs cycle, right, to pick up more and come back later. And what we're eventually doing is getting this high concentration of H plus here, which we will then later use for chemiosmosis. Okay. So in chemiosmosis, the H plus is going to flow through the ATP synthase and spin the ATP synthase around. And if maybe this thing is working now. Not really. Doesn't.
Brain drives FO to rotate.
I want to shut up. No, it really doesn't like me. Maybe I'll just post it later. Anyway, it really clearly shows like how the thing spins. So you can see like ADP and P getting shoved together. I don't know. Oh, well. It's not loading. Yes, sorry.
What's the purpose of the hydrogen ions again?
It's like in plants, you know, when we create that fake gradient of ions. It's sort of along those lines. So we're like putting them all over here to eventually let them diffuse back this way in order for us to make ATP.
So the hydrogen ions came from the electron carriers.
Correct.
The 10 from NADH and 2 from FADH, and nothing through the drawing just now. You have an ETC, right?
Yeah.
There's nothing on the C, right?
Nothing on the C means. What do you
The
mean?
red chunk, is it?
Sorry, say that again.
The drawing on the iPad, you have an ETC and the ATP site synthase, right?
Yeah.
Is there anything on the C? Because I didn't see. And then
C can also, it might also pump some protons. Nothing gives its electrons to that third protein though. They only give their electrons to the first two.
wait, so basically, hydrogen ions got pumped to the inner membrane space, and then they just get pumped back through the ATP synthase. Wait, it's an enzyme, right?
It is an enzyme, yeah.
So how does it get pumped back?
Okay, first it's not pumped. They diffuse, right? They're going from high to low concentration.
Diffuse.
Yeah, and they're just going through because it's like a channel. It's like a channel protein that's also an enzyme.
Okay.
Yeah. So the H plus goes through this channel because it fits, it works, it's accepted by this protein. So it's allowed to flow through. As it flows through, it powers the bottom part to turn like this. And this is why if that would just work. Here we go, come on. It makes this thing spin. Yeah, so it like turns like this and it shoves ADP together with P to make ATP.
Oh, okay.
Okay, so the hydrogen flows through. Again, like think of the hydrogen like a waterfall. So the hydrogen flows through and then there's like a wheel that helps to spin this bottom part around. That spinning part takes ADP, PI, puts it in like an annoyingly uncomfortable position where they just get shoved together.
And then you said the hydrogen forms water?
Yeah, coming. Let me get to that part.
Oh, okay.
We kind of jumped ahead a little bit. Okay, so okay, ATP synthase makes ATP. So what we see here, so there's the H plus flowing back through from high to low concentration, right? It's basically just diffusion, although we call it chemiosmosis, whatever. It's something moving from high to low, a chemical instead of water in this case, across a membrane. That's the reason why we call it chemiosmosis. But now we have to we got to get rid of these H plus ions, right? Because if we have a lot here, or we start to balance out between intermembrane space and matrix, then there's no need for flow of H plus, which means ATP synthase stops spinning, right? We have to keep it spinning to make ATP continuously. So we sort of have to like do something with these H plus. We have to clean them up somehow, right? To maintain a gradient difference across this membrane. So how do we clean them up? Okay, we just mentioned that. How do we clean them up? Here, you have that oxygen that we've needed this whole time, right, for aerobic respiration. This is where it becomes most important. It hangs out at the end of the ETC, right? It accepts 2 electrons and 2 H pluses, an O2. So this is to answer Yushwan's question. An O2 molecule would split apart, take 2 H pluses and 2 electrons, which gives you H1, H2, plus half an O2 is just O. So you get 2 molecules of water. So for every 4 H pluses you can grab, and for every 4 electrons you can grab from the chain, you can make 2 molecules of water. What does that do? It cleans up. I like the term here. The term here they use is they say to mop up the excess H plus, right? So it's like O2 is like a sponge. It's going to clean up the hydrogens, clean up the electrons, make water, and now you have low H plus here, right? Because you've cleaned up the H plus by making it a part of water, which means H plus here can be high.
Okay.
So that they keep going this way. Yushin, did that answer your question?
Yes.
Are you sure?
Pretty sure. Yeah, I think I'm good.
Okay. So again, lots of summaries, lots of recaps that you can check out that will essentially like go through this whole thing. Some in maybe a bit more detail than is necessary. For example, I can see already here that they're talking about the names of these proteins and stuff. I think the McGraw-Hill one is a good level. They don't get into too much detail. So if you need it, check this one out. They have 2. I sent 2 links yesterday. I realized there's another one in addition to this one. They're both good. And all that remains is what I said 5 lessons ago, which is to, this is just another summary of how it all works with a little bit more detail of like the names of these proteins, like complex 1, complex 2, not really what we're not interested in that, okay? The process is more important in this case. What we said, so here's your summary, but I'm just gonna skip that. You've got some nature of science, okay, yay. It's hard to change people's minds when they believe something for their whole life. This is how science works, okay? People have an idea that they've taught it in school, they've taught it all through their life, and then when someone comes up with something better, that they know is better, it's really hard to convince people to change their minds. It's just one of those things we call paradigm shift. We've got some questions you can try. This is what I was saying we would come back to, okay? Is now that we know all the processes that are going on, we can annotate our first drawing that we did a long, long time ago with what goes on in those places, okay? So now we know that, for example, this inner membrane, all over this inner membrane are electron transport chains. So we showed 1, right? But you can have this happening like hundreds of millions, maybe not millions, hundreds of thousands of different like electron transport chains all over this inner membrane. So therefore, why is it important that it's highly folded into cristae? Because now you have more surface area, which means more length of membrane to have more ETCs, which means more energy, right? Why is the intermembrane space important? Well, because it's a really, really small space, so you can build a hydrogen gradient really fast, right? So here you've got the details that I would add to your drawing from the very first lesson. Pause, breathe. What do we need? Anything else that needs a recap? Really?
Daniel,
Yeah.
is it like
Go.
the first, like the first protein has the NADH and the second one has FADH, or?
Yeah, let's keep it, we're going to keep it that way. So the NADH gives its electrons to the first protein in the chain, and FADH gives its electrons to the second one. So you can think of it as like the electrons from NADH actually travel further than the ones from FADH. If you read some of the stuff like that goes into more detail, they will say like, therefore, like NADH yields more ATP. And the reason they're saying that is because those electrons travel farther, which means they are responsible for pumping more H plus, which then will eventually come back. So they're saying like, if you wanted to compare the two, NADH is actually better at generating energy at the end, but it's only because FADH joins a little bit later on the chain, so it doesn't pump as many H pluses because it only moves like from from the middle to the end, whereas NADH is over here, which means it has more time to pump hydrogen ions. But that's just it's a way to like compare the two in terms of which one's, let's say if you ask which one's better at generating ATP, but most likely you won't be asked to do something like that.
Oh, okay, what about the third protein? What happens to that one?
It's
because like you drew ETC.
Yeah, so it's also pumping, but it's not accepting any electrons.
Okay, so the electrons stop at the second protein, like accepting electrons stops.
Yes, yes, giving away, yeah. So for our like simplified model, we can think of it as if there's 3 proteins, NADH gives their electrons to the first one, and those travel from 1 to 2 to 3. FADH gives its electrons to the second one, they travel from 2 to 3. All the electrons, when they get to 3, are going to get grabbed by oxygen, and that's basically it. And at all 3 of them, hydrogen can be pumped across.
Okay, thank you.
I'm gonna try one more time for this stupid thing to work. Oh my gosh, it's working.
Catalyzing.
No, piece of crap. Oh well. I think if I turned on my VPN, it would work, but then I'm not sure what would happen to the Zoom. So I'll just post the link to this later. All right. Well, I guess looking ahead, we'll see that a lot of these kinds of concepts like come back when we do the HL part about photosynthesis. So like these like things like electron transport chains, they and ATP synthase, these things are there again, even in photosynthesis. So this isn't like a one-off. So we kind of learn this and then we'll reuse this similar information again when we see how, for example, the light independent and light dependent reactions, how we are able to make glucose out of light energy. A lot of the same concepts are going to come up again. But other than that, that is the end of the gargantuan topic of cell respiration. How do we feel?